What's going on, everybody? Welcome back to the show. This is the Science Dispatch Podcast. I'm Cameron English. That guy over there, handsome guy, Dr. Chuck Dinnerstein, Director of Medicine. Always excited to see you, Chuck. We have a special guest with us this week, Dr. Joshua Bloom, concert pianist, chemist, all-around sharp guy, maker of really funny jokes. It's good to see you, Dr. Bloom. How are you?
I agree, Dr. Cameron. I lost the screen view.
It's okay. We can see and hear you, and that's what matters.
Yeah. How did I manage that?
I don't know. We were just talking off air about a silly faux pas that you committed, and it boggled my mind because you're such a brilliant guy. The chemistry you understand and explain, it's beyond most people, and yet here you are. You're like, how does this camera thing work? In any case, Josh is going to try to figure out his camera, and we're going to do the intro to his story here, which is...
I can see you now.
Okay.
Can you see me?
I can,
Yes.
And I'm grateful for that. I'm grateful for that fact.
Isn't that clever?
We're having a good time here. We turn technical difficulties into bad jokes. That's what we do. Josh, your story is called SU212. A potential cancer drug missed its target and found a better one. This is a rare and encouraging story we get to talk about on this podcast, which is often focused on difficult news and challenges that aren't easily solved. But Josh, I'm going to read the introduction to your story, and then maybe we can talk about some of the specifics here. So you write, sometimes big breakthroughs in science come from very small changes, curiosity, and more than a little luck. A new study from researchers at Oregon Health and Science University shows how swapping a single atom in a known compound created an experimental cancer-fighting molecule with a completely different mode of action and improved properties. Okay, so Dr. Bloom, 30 seconds to a minute, just a quick elevator pitch in as simple terms as you can manage. What did they do here? Were they looking for something different and they went, hey, wait a minute, there's a better way to do this?
Well, they were doing how this is still drug discovery, old-fashioned. They used a tried and true method called bioisosterism, which is a fancy way of saying replacing an atom or a group in a molecule to see if you can give it better properties, better solubility. It's done all the time, and there are drugs out there that have succeeded because they were able to change the properties to make them more soluble, less toxic, or whatever.
Is there a rule book that says, if you swap here, you will get greater potency, greater solubility, or is it pretty much a crapshoot?
Potency, potency, what you do is they model it, and they see that it fits a receptor or whatever better, and then they use the modeling. They pretend that it's they use the modeling to discover it, where it's always backwards. They always make the discovery and then rationalize it with the computer assisted. Not always, but
So it is a crapshoot. They just kind of fool around, and whatever works is.
Yes and no. They did one change in this molecule that is a fairly obvious one, and they took a drug called podophyllotoxin.
Oh yeah, of course.
Which is a toxic cancer drug. It's used, and they replaced a carbon atom with a nitrogen. It's a basic nitrogen. And they also attached to the nitrogen a small carbon chain with an alcohol. Both of these things make the molecule more polar and more water soluble. That you could predict with 100% certainty. Whether the activity, toxicity, metabolism is better or worse, you just have to try it and find out. And there's still, despite the discussions that AI is going to revolutionized drug discovery. Well, that's all fine until they run into something with an unexpected toxicity 2 years into the project.
So tell us a little bit more about what this drug actually does. So the change they induced, it makes the drug somewhat less toxic and actually makes it more efficacious, I think, if I'm reading your article right. Can you talk a little bit about that?
Keep in mind it's just in mice, but they're using, they have a tough target, and that's triple negative breast cancers, the kind that you can't treat with aromatase inhibitor or HER2. When breast cancers don't have a target like that, they tend to be much more serious. So they, the oncologists pile up the drugs to try and knock it out, and this illness, and just the efficacy is not good enough so that a dose can be given that isn't really toxic, because these are cytotoxic, so they kill cells.
So the side effects are additive, especially as you add more drug to the cocktail, because this triple negative breast cancer is the high hanging fruit. The low hanging fruit is well treated. This is the remaining kind of breast cancer, say, that continues to be problematic. It has not been converted to a chronic illness.
So what's the goal with something like that? You just hope it kills the cancer before it kills the patient, or I don't mean to sound snarky or anything, but if the drugs are that toxic, you're just kind of running a calculation. You're like, well, it'll take out your cancer before you die, and so is that the situation that you're trying to get away from with a drug like this that they're experimenting with?
Yeah, it's very easy to kill cells. But, a couple of decades ago, you started getting targeted therapies for HER2 and, genes that block a certain function of a gene that promotes the cancer. And so I don't want to make light of it. These were really important discoveries. But what's happened in a lot of cases is the immune modulators, or whatever type of biologic is given, and there are plenty of side effects for that. And then they add the chemotherapy on top of it to make it work better. So.
Interesting. So even with the gene therapies, you still need chemo.
Not always, but sometimes.
Sometimes. Okay.
Chuck, you probably know more about this than I do.
No. I'm going to stay out of that because I don't want to get myself in trouble. But it seems to be, I think, that the interesting piece was the serendipitous finding that with the chemical change to solubility, there was also a mechanistic change, that the target abruptly changed.
Yes.
And much more efficacious, I gather.
Right. So this is called luck. And if you don't have luck, you're never going to discover and develop a drug, because it requires an enormous amount of it, no matter how good you are. So what they did was try to make it more soluble, and it turned out to be more efficacious and less toxic in mice models. Now, that's fantastic. The question is why? Because a lot of these cytotoxins are mitosis inhibitors. So they kill cells, cancerous and otherwise. But taking podophyllotoxin and putting an ASA group on it, that's what they call it, ADA podophyllotoxin, it did achieve the solubility changes that they were looking at. But then it turned out it binds to an entirely different place. So by not binding to the place where mitosis is inhibited, you take away that particular form of toxicity, and that's a pretty bad one. And it bound to another site on the triple negative cells that is involved in energy glycolysis and energy generation. That's really lucky, because no one's ever going to predict that. So you got this, you got a really toxic molecule that binds, prevents the mitotic spindle, and this is a lot of chemotherapy it does this. One of the first chemo drugs, vincristine and vinblastine, they were mitotic spindle inhibitors. Those were from the 60s or 70s. Yeah, so you want to get away from that, and they did with some luck.
Is this new drug much more specific to the breast cancer cell, or is it interfere with glycolysis across a larger spectrum of cells?
Well, sort of both. It turns out that this breast cancer cells are highly dependent on having large amounts of energy, and that's generated through glycolysis. So if you slow that process down, you really slow down breast cancer tumor spread. It just so happens that, for reasons that I don't want to go into and don't entirely understand, breast cancer cells are more sensitive to this type of mechanism than not only regular cells, but other types of cancers. So very fortunate and
double lucky.
Double lucky, yeah.
So what would be the next step? Presumably if this goes anywhere, it's 15, 20 years out. But what would the next step be? Would you do studies or bigger studies with more animal models? And then if those are successful, then go on to some sort of clinical research to see if this works?
I'd have to see the whole tox package in mice or rats or maybe even in other species. But and normally you would do some structure-activity relationships. In other words, modify different parts of the molecule and see if it's better or worse. Not with this. If this does even close to what's advertised, I get this thing into the clinic real fast. This is a huge problem. And if you could really develop a less toxic, more effective breast cancer drug, then bingo, you really hit something. So you have to be careful because this is in mice, but it does have relevance to humans. It's just not perfect.
Well, I mean, over the last several years, from both administrations, we heard a lot about this cancer moonshot proposal, and then under the leadership of RFK Jr., Health and Human Services Secretary, still blows my mind that that's a real thing that I have to say. He's made a lot of claims about the chronic disease epidemic and all these things we have to do to make America healthy again. Presumably, with the right support from the federal government, you could accelerate the development of drugs like this. Do you suspect that is even possible, or is this going to languish? What does your forecasting tell you, Dr. Bloom?
Well, academic research is, it's often funded by federal grants from the NIH. And in the absence of it, some of it's going to go away. Now, I don't know how these guys funded this, and it's easy enough to find out. Whether Kennedy thinks that chronic diseases are, I don't know, come from what? I think I'm werewolf, so I don't, I know, what does that mean? I just had to get that in, sorry. This is, I don't know, this looks like a super lucky, unintentional moonshot. You'd never try to do this. as a moonshot. It's traditional medicinal chemistry, which, if this continues to work, it will be, an enormous advantage. But
So it sounds like the whole AI and drug development may accelerate some of the modeling, but will ultimately be rate limited by serendipity, which seems to be a big driver.
Right. What the 29-year-old billionaires don't realize is that drug discovery gets tough as you get further along. Like, it's pretty easy to design a molecule to inhibit an enzyme, and that's been going on for decades. So they design many more molecules to fit or inhibit an enzyme, or ones that are going to be more soluble. They can do that, or ones that are going to have a different conformation. Well, that's fine. That's very useful. But when you give it to a dog and the dog drops dead from some process that you could have never predicted, then you got a problem. So this is why drugs don't just go from animals to humans seamlessly. It doesn't work that way.
All right. So
there's the clip for social media. Okay, so in sum then, this is some good chemistry mixed with a whole lot of luck, and provided that the next stages of research are equally successful, this could be a promising development. We're just not far enough along to say that with any confidence.
I agree, but if this turns out to be real, then you got a new target. It's called enolase 1. So you can screen bazillions of compounds against enolase 1 and slow down breast cancer cell growth while leaving most other cells alone. So this could go in any number of directions. I thought the most interesting part of this was how they saw that a very similar molecule no longer binds to the mitosis site. It binds someplace else, and they see the interactions that make it do so. And it all makes sense. I mean, you can see why it fits there. I don't think you'd ever predict that it would fit there. But,
that seems to be another area where they've been trying for a long time that they still have not got their hands fully around it, is translating molecular structure into that 3D conformation. There's just no way to adequately predict that this particular structure will form this or that particular shape.
Not during the time when I was doing drug discovery. You could predict the lowest energy conformation of a given molecule. That was pretty standard. But to predict that it would bind in a site that's not active site and inhibit a different process, I don't know. It's pretty far-fetched.
Well, hopeful. We can't say sarcastically that we solved another of the world's problems, Dr. Dinnerstein. But this is actually some promising research. And I did just look it up. They have looks like they have some foundation funding, and they have funding from other governments besides our own. So this is not going to be, at least at this stage, contingent on whether the overlords at HHS deem this worthy of public funds. So that's good. One more time, the article is called the article is called I have kids, everyone. It works really well for your focus. Once again, the article is called SU212, a potential cancer drug missed its target and found a better one. That is on acsh.org. While you're there, click the subscribe tab. Give us your email address. We'll send you the stories we publish. The ones that you read are usually the ones we talk about. We're on social media at ACSH.org on X. Dr. Bloom has his focus group generated name. It's at Josh Bloom ACSH.
Clever, no?
It's very clever. I do enjoy that. But you are a good follow. There's lots of good insight. So follow Josh. You can follow me. I'm at CamJEnglish. We are on Instagram, Facebook, YouTube, ACSH Matter of Facts, hosted by our colleague Anna Delaski. Little 1-minute videos explaining science questions that are floating around the internet these days. So go check those out. Thank you so much as always, and we will see you next time.